I think we can start.
Please, of
Should
course.
I?
Thank you.
Yeah,
Sure.
I'm sharing my screen. So today I will be presenting about causal inference. So basically, previously my idea was that I will present the jargons about causal inference and then try to give some brief overview how it is used in drug discovery. But later it turned out to be too big of a slide. So uh, today for this presentation, I will be explaining the causal inference and the jargons used in causal inference, and later in another presentation I may try to present a paper about drug discovery, how drug discovery uses causal inference to um, create drug reposition. So yeah, so for understanding causal inference, yeah, it will be better if we start with randomized control test. So let us give a situation. So assume that you have a e-commerce website and you are trying to increase your sale. So what you can do is that you can send your users mail about giving some promotional offers and informing them about your products. That may improve the purchase conversion. But how much improvement this will create for us? Will it be significant? So for this type of test, a very well-known test is known as A/B testing. So this is also A/B testing. Actually, what does it do is it does the randomized control test. So what is the idea is from the group of from the total population, you have to select a group of population. So for our case, some users randomly, we can select some users from all the users in our e-commerce website, and then later we can divide the population into two groups. Um, the groups can be named as group B and group A. So the group B can be called treatment group and group A can be called control group. The third step is we send email to the treatment group. The email will contain promotional coupons and information about your product, and we keep the control group as it is. We do not send any email to them. And then the fourth step is to monitor the conversion rate. So the if the situation is as we expect, then the treatment group should increase purchasing your product, and control group should not be very motivated to purchase your product. So the fifth state is taking a decision. So you can evaluate the conversion rate, where the email is useful. So this is actually the randomized control test. Yeah, Unup, do you have any questions?
Sorry for interruption. I'm just wondering if the control group don't get any email. So how are
Mhm.
we uh, deciding that the emails were useful or not? We're not sending them anything, right? So
Yeah, so the treatment group is getting the email. So if the conversion rate for the treatment group increases, then we can think that the email was useful. And for the control group, we are not sending anything. So the flow should be a normal flow. I mean, a normal flow that comes to your website.
oh, okay,
Does
okay,
that
yeah.
make sense? Yeah, thank you. So a visualization for this randomized control test can be shown here. So the first row will show the selected population. Say that you take 20 people from the whole group of users that you have. So for the first 50%, you can think it to be a group A, that is the control group. And the second 50%, you can think them as group B, that is treatment group. So for control group, you are not sending any email, and for treatment group, you are sending them promotional email. So you can see that the control group from the control group, there are 20% conversion for your product, and for the treatment group, there are 40% conversion for your product. So this can from this conversion rate, we can say that sending the email was somehow a bit useful. So one thing that we need to notice that we have said very strictly that the population should be randomly distributed. Uh, so while selecting the population, it should be random, and while dividing them into two groups, it should be also normal, randomly distributed. So what is the problem with randomized control test? So randomized control test is unethical, sometimes unethical in medical experiments. So you for randomized control test, you need to conduct experiment on real data, but it is not always possible for medical experiments. So what we need to do is do some inference with prior data. So here comes causal inference. So for causal inference, we need to solve the same problem, but we have to now
Noman, I
consider.
had a question actually in the previous slide.
Mhm. Yes, sir.
I thought that's what the medical industry does, the randomized control test. You have a case and control, and then you give the medication to your case population, and you don't give the medication or the placebo to the control group, and then you do the study, right? Whether the condition improved or not, was
So
or not.
yeah, um,
So that's where I'm confused.
like,
Like this slide is claiming this is unethical in medical experiments.
so this is sometimes unethical as far I do understand, because
So, I mean, just to like fill in here, let's say a drug, it's not like FDA approved yet, or it's not like past the first few clinical trials, then
I see. Okay.
yeah.
Okay. So this is there is a proper protocol of like going to the first stage, second stage, I guess, and then the third stage is probably this randomized control test, right?
Yes, that's what I understand. Like it goes through, let's say, some lab test and then on mice and then based on like donors. And yeah, so I mean, it's true that in general, we just can't do a randomized control test for any drug.
Right. Okay.
yeah, that maybe is the case. So from that limitation, there comes the causal inference. So when we do not have the capability for doing the experiment, so for example, um, the previous example for randomized control test was sending an email, but if that was to uh, uh, you make a billboard and posting it in the road, that would be very expensive. So in such cases also randomized control test will not work. So in that cases, what we have to do, we have to rely on past observation. So there comes the causal inference. So the past but so for example, here we have the same kind of situation. We have some treatment group and control group, and we for treatment group we are giving them vaccine. Let's say after giving the vaccines, if 70% population are cured, and the control group, there is no vaccine, there is a placebo, and 30% patients are cured. So you can say that the vaccine works pretty well, right? But there is a catch, because the data is past data. We do not have control over the quality of the data. So let's say that the treatment group has an average age of 25 and the control group has an average age of 37. So as these data comes from our previous observation, they are messy, and we do not have control over this age. So yeah, that would be a problem. So here comes another jargon for that, that is confounder. A confounder or a confounding variable is a factor that is associated with the dependent variable and also the independent variable. For example, here the outcome is if the patient is treated or not. So um, age uh, age is a confounder here that that we have to consider, and that is related to the outcome also and the treatment that we give. So we can think it that like a way that if a person is very young, there is a high chance uh, he will be fine from a flu very quickly, and the treatment may work for elderly people very well. So there is a effect of this confounding variable one on both the treatment and the outcome. So yeah, so for causal inference, there are some assumption. The first assumption is that causal Markov condition, as we have seen before, there is a confounding variable that has effect on cause and effect both, and cause has effect on cause can change the effect. So this will be a directed acyclic graph. Another assumption is stable unit treatment value assumption. So for this assumption, we think that the treatment will not vary. So say that for a person, if for two different person, the treatment the treatment level will be equal. So there is no power for a vaccine like that. So there will another stable unit treatment value assumption is no interference. That is, one unit treatment will not influence another unit. So the people from the control group will not influence the people from the treatment group. Another assumption for causal inference is ignorability. That is, we will not consider any unknown confounding variable. So if we have any unknown confounding variable, this causal inference may not work properly. So we need to know all the confounding variable for us. So let's see how a causal inference, how we can calculate causal inference for the previous case that is giving a vaccination. Say that we have 5 patients, and they are divided into treatment group and the control group. So for example, here you can consider that Noman, Zing, and Konstantin, they are in treatment group, and Sam and MCS is on the control group. So you can see that in treatment group, Noman is after giving the vaccine, Noman is treated, Zing is treated, but Konstantin is not treated. And in the control group, you can see that Sam is treated uh, without giving any vaccine, and MCS is not treated without giving a vaccine. So that are the situations in the table. And you can see that here is a column for age. That is our confounder in this case. So for founding the causal effect, we need to find the counter examples for each of the cases. For example, here we can see that no one has been freed.
So
Right.
Noman, I'm sorry.
Yeah.
So, how did you deduce that age was a confounder in this case? I
So
still get the point. Sorry.
yeah, so for causal inference, we can have many confounding variables. So
Mhm.
in this example, we are thinking that age is a confounding variable that can impact the vaccination testing. So um, you can think that those who are younger, they are much probable to get treated by themselves, without any vaccination. So age can be a confounding variable for those drug experiments. Does that make sense?
Yeah, but I guess I get your point. So there is not enough data in this slide to make that, but I understand the point. Okay.
Right. There can be many confounding variables actually. Their age, their size of their kidneys, and many things. So for making the slide easier, so there is only one confounding variable here.
Sure.
Yeah. So yeah, so for finding the causal effect, we have to find the we have to find the counter examples for each of the observations. So for example, here we can see that after Noman is in treatment group and after giving the vaccine, he is treated. But what would happen if he was in control group? So uh, if he was in control group, will he be treated? So we have to find the counter examples for each of the observations. So how can we do that? So for finding the counter examples, we can run some machine learning algorithm or some matching algorithm. So for example, you can see that in the first two row, Noman and Sam has similar age. They're of age 25. So we can see that Sam is from control group, and without the vaccine he was treated. So so for Noman also we can give it a 1. So here we can see that Sam was in control group and he was treated without the vaccine. And so for Noman also we can impute um, this value here. So in this way we can populate the counter examples for each of the observations that we have. So after populating the observation, we can actually find the causal effect. So the causal effect is the difference between the two outcomes that we have. So for example, for Noman, the treatment outcome is 1 and control outcome is all 1. So the causal effect here is 0. So the causal effect for using the vaccine is 0 here. For Sam, it is 0 also. So for Amcius, we can see that it is 1. Zing, it is 1, and for Constantine, we can see that we have the opposite outcome of vaccine. So without uh, so here it should also be a counter example. So yeah, Constantine was not treated with also giving the vaccine. So this is a counter example. So in this case, um, uh, so in this case also it is -1. So we can find the causal effect for each of the observations, and after that we can find the average treatment effect. So average treatment effect is the sum of the treatment outcome minus control outcome divided by all the observations. So we can see that the average treatment effect for this drug is this vaccine is 0.2. So that is not too much. So we can say that the vaccine is not very useful um, for this population. So, however, there is another thing that is conditional um, causal treatment effect. So, if we condition over the age, say that the third row, we can see that average treatment effect for those who has age over 32. So we can see that those who has a age over 32, for them the causal effect is 1 for all the cases. So in the third and fourth row, you can see that the causal effect are 1 and they have a age of 33 and 34. So we can say we can find a conditional treatment effects. So if we condition by age of greater than 32, we can see that the causal effect there will be 1 that will indicate that the vaccine is very useful. So we can understand that confounding variables can play an important role over finding if a treatment is useful or not.
Noman, now I have a confusion. Can you
Yeah,
go back to the table? So
yeah,
I
sure.
mean, I maybe lost this part. So for one patient, how can I get both the treatment outcome and the control outcome? Can you like review the way this experiment was designed?
Yeah, so for one patient, we actually have this observation data. So so for each patient, we either have the treatment outcome or the control outcome, right? So
Yeah.
we have to find the counter example to find the causal effect for that patient. So for Noman, we have the treatment outcome, but we do not have the control outcome. So what we can do is we can we can design a machine learning algorithm or a matching algorithm. So for example, here we can consider a matching algorithm. So we can think that people of similar ages will have same kind of outcome.
I see. Okay.
Yeah.
So
So
those empty blocks, empty cells, they're like inferred using some
yeah.
model.
So
Okay.
they're inferred using some model. So if we consider matching model, we can think that Noman and Sam has similar ages, so they will have similar type of outcomes. So
Okay.
Noman will have control outcome 1 here. So yeah, that way we can find the counter examples for each of the observations. So in this way we can populate the blank column. So they're shown in color cyan here. So yeah, so after we populate them with some machine learning algorithm or some other model or some matching algorithm, so we can get this causal effect.
Makes sense. Thank you.
Yeah. So these are some jargons about causal inference, but how they are used in drug discovery. I will not go through very majorly about this effect, but I'll give some brief overview of it. So for drug discovery, we have some drug and some drug and some diseases. So for row 1, we will see that there is D1 to DX. So we can think that they are drugs, D1 to DX. And for second row, there are some T1 some targets. So the target can be thought as some proteins or something like that. So the targets are from T1 to Tn, and there are some pathway for sorry. So there can be some pathway for related to each target. So we can think that drug 1 triggers target 1, target 2, and target 3. So so there is a cause and effect relationship between drug 1 to target 1, drug 1 to target 2, drug 1 to target 3. So for each target, there are some pathway related to each of the targets. So for example, we can think that target 1 has pathway P1, target 2 has pathway P2. So each pathway also can trigger some genes. So for example, we can think that pathway P1 can trigger gene 1 and gene 2. Pathway 2 can trigger gene 2 and gene 3. And each gene expression, triggered gene expression, can cure some. We can think that these gene expression are some upregulation or downregulation due to triggering of those pathways. So each upregulation or downregulation of gene can be related to curing some of the disease. So for example, here we can think that gene 1 and gene 3 can upregulation or downregulation of gene 1 and gene 3 can treat disease S1, and upregulation of gene 2 and gene 4 gene 4 and gene K can treat disease SY. So like that, we can create some causal network. So this is not a simple causal graph. This is a causal network. So with this causal network, we if we have, so for example, we know the behavior of some of the drugs. So we can think that the YOLO lines indicates that these are known from previous experience. So we know that drug 1 triggers target 1, target 2, and target 3. We know that drug trigger 1 activates pathway P1. We know that pathway P1 activates G2, but we do not know that pathway 1 activates G1. I mean, we know this, um, but for a certain drug, this is not observed. So yeah, so if we have those relationships, we can create a weighted graph here, and we can find the maximum likelihood for each pair of drug and drug and diseases. So in this way, we can find some drug and disease pair that was previously not known to be useful. So we can think this we can think this in a way that previously we didn't know that steroid drugs were useful for treating COVID patients. So yeah, that was a new discovery. So this is called drug reposition. So a drug is known previously, but there can be new use cases for those drugs. So yeah, we can create a causal network like this, and we can find drug reposition between two different drugs. So yeah, I will try to explain this paper later in another presentation. So for now, that is all from my side. If you have any question, feel free to ask.
Yeah, this was awesome. So I'll start with a clap and then others can ask questions. I mean, of course, we are interested in this drug repurposing thing because that's the more practical thing to do. And there is a technology called Perturb-seq, which is not necessarily a drug-based so you can imagine that every drug, it causes some kind of perturbation in stage 4, in level 4. It impacts some gene. Now that perturbation may be for the good, or like it may even worsen something. That's a different thing. But it's more about a drug causing some perturbation. And in Perturb-seq, it's not a drug, it's a gene that's like turned down or like made more active. And we try to, like, Perturb-seq tells you that which genes have like the ability to impact which pathways. So Perturb-seq is a way to discover pathways that are not known yet. So that's one type of data we are trying to model, which again comes very close to spirit in drug repurposing. Yes, Perturb-seq. Anyway, so I look forward to your next presentation when you talk about that paper. Which paper is this, Noman? This multi-layer concept graph?
So this is a very old paper, but they claim to be the first one to use causal inference in this drug repositioning. So they claim that previously they were done using creating some similarity network. They have introduced this causal inference. So for this reason, I was interested in this paper, though it is very old one.
Mm-hmm. That's fine.
Yeah.
So, Noman, I had a question.
Yeah, sure.
If you go back to the slide, yeah. So here, what are the targets? Can you give me an example of a target? What does it mean?
So the underlying biology I'm not sure about that. Maybe Sami sir may give us some understanding about this. So they have given an example of it. So here is the example. So they say that they said that through this they have found that a drug that is called bixarotin that was previously used in treating cancer. It has a new pathway that indicates that it can be used for Alzheimer disease that can clear and it can clear the alpha beta amyloid from the brain. So it says that as a drug target, this drug bixarotin can activate this R cross R target. So R cross R they says that this is retinoid cross receptor, and this retinoid cross receptor can trigger the pathway L cross R. So L cross R they says that liver cross receptor. So this liver cross receptor can also
Okay, I think if you go back to the slide,
Yeah.
so Sami can perhaps confirm. If you go back to the slide, so the target itself is another gene, right, Sami?
It need not be. For example, a drug, it can target some other molecules in the cell. A drug, it can sometimes help, let's say, make the channel pores in a cell wider or narrower. Like that, there are channel pores, like tunnels, through which molecules like sodium and calcium, magnesium, they go in and out of the cell. And a drug can even like target those pores. So I
Mhm.
thought that target here is more of a generic term. It can be anything that
So it
actually
could be like multiple physical things, right?
exactly, yes.
But then that the outcome of that drug interacting with the target is that,
I
So
lost
maybe
your voice.
sorry.
I mean I just lost your last few
Okay,
sentences.
so I'm trying to rationalize. So the drug combining with the target triggers some sort of pathway, right? And then all the genes within that pathway are getting activated, or they are getting upregulated or downregulated or whatever, and then that can treat the disease. This is what this thing is showing, right?
This is what this is showing. That's correct.
Okay.
The other thing is again there are these I think blood pressure drugs. I think they call them beta channel blockers or sodium channel blockers. I don't
Beta
know.
blockers. Yes.
Exactly. So so they do not necessarily target the genes. And what I'm trying to tell here is maybe by targeting some other molecule in the cell, we can relieve the symptom. We can like make it look like that the disease is there no more. Does that really cure the disease or fix the issues at the very root level, at the gene level? That's not yet understood. So what this shows is like a very elaborate view. But in as much as I know, many times people just think of like finding a target that can cure a symptom. If that symptom is like just cured in the face value, or is that like going all the way down and like fixing the genes, etc.? That's not always well understood. And typically they don't even claim the drug being able to fix things at the like root level. I don't know if I'm making any sense. Okay,
Sure.
maybe not. So
No, no, sure. I understand.
yeah. So again, I mean this beta blocker thing, right? I mean maybe I am predisposed to high blood pressure genetically. I have like some genes going in the wrong direction. By going, I mean being expressing too much, or not expressing at the sufficient level. The beta blocker probably won't fix those gene expression values. The beta blocker would do something at level 2, and then try to like keep the blood pressure steady. I mean, I guess I'm saying this for the other ones. I mean, but that's what I meant. So CRISPR and those technologies that um,
Mhm.
RNA inference, RNA interference, RNAi, they call it RNAi. They actually go all the way down to like level 4. But the problem
This is
is
from the genes, right?
exactly, yeah. But the problem is knowing the dose level or making it very specific to, let's say, gene 3 and gene 4. Those become challenging. Like, because when I'm targeting T1, yes, I'm targeting T1 and I don't care about like anything more specific. But if I need to like specifically target G2 and G3, that's like a lot of engineering.
Okay.
Any other questions?
So, Noman, I have another question from this slide. Like, do the different colors on the causal links represent? like the orange color and the black color?
Yeah, so the orange color indicates that these paths are known for us. So from the previous data, you know that drug one treats
Mm.
disease SY. So for disease SY and drug D1, so there are some paths that are known that are indicated by those orange links.
Mhm.
So yeah, so from those are known, so some data are known for us. So we are trying to find the unknown thing. So what will be the effect of drug D1 to disease S2 and drug D1 to disease S1, like that.
Okay, okay.
Does anyone else have any question? So maybe not. Let me stop the recording.